I always like that it tells me that just so I can be sure.
Ethan, will you need our presentations as well?
That is up to you, actually. So that goes for all of you. If you are willing and wanting your presentation slides to be shared on our website, by all means, just send them to me as a PDF after the webinar, and I can include them below the video link. Completely up to you. Some people prefer not to. They like to have the auditory explanation going along with the slides, but if you'd like to have them there just as supplementary info, that's totally fine as well.
Thanks.
Yeah, just let me know. We can talk about it afterwards. It usually takes a couple days to get the webinar uploaded to the website and have Zoom process and everything anyway. So yeah, there's a bit of time to decide whether or not you want to do that. Hello, good morning, good afternoon. For those of you who have joined us so far, we'll just give it a few more minutes, but just wanted to say welcome. Thank you for being here for our 8th and final webinar of 2022. It's been a busy schedule for us. We'll just give a few more minutes for a few more attendees to filter in, and then we'll get started. Thank you. Alrighty, I think in the interest of everybody's time here, we can dive in and people may continue to filter in, that's all right. Thank you everyone for joining us today for CGCN's 8th and final webinar of 2022, as I had mentioned. This today is another in a series of grape and wine cluster research updates that we've been holding throughout the year. This is the 5th one of those. And today's focus is on the theme of optimizing the quality of Canadian wines. My name is Ethan Churchill. I am the project manager for CGCN-RCCV, and today I will also be serving as your moderator for the webinar. We're joined today by 4 wonderful guest speakers. We have Dr. Debbie Inglis, Dr. Gavin Kernaghan, Dr. Lihua Fan, and Dr. Belinda Kemp with us here today. Just a couple administrative notes before we dive in. I'd like to mention primarily that this webinar is being recorded and it'll be posted on our YouTube channel and our websites afterwards if you wish to either view it again or share it with colleagues who couldn't make it today. Also, we will be holding a question and answer period at the end of all 4 presentations today. So if at any point any of our attendees have questions for our presenters, you can type them in using the Q&A function at the bottom of your Zoom screen, and we'll address all the questions at the end of the presentations. Also, speaking of the presentations, during the time that someone's got a screen being shared, if you want to change the size of the presentation relative to the camera view, if you hover your cursor towards the right side of the presentation screen, you'll see a little double line that you can use to resize that image to make it a little bigger or smaller as you wish, depending on your preference. Just quickly before we dive into the first presentation, I'll just quickly go over a little bit more about CGCN and about the Grape and Wine Science Cluster for you here. So for those of you who have attended a few of our webinars now, this slide will look really familiar, but maybe if this is your first time joining us, I'll just go over some of the broad details again. So our application for the Agroscience cluster was submitted to Agriculture and Agri-Food Canada in January of 2018. The cluster activities were built upon past research in each province to create a more national coordinated effort to address a variety of key challenges in the grape and wine sector. Our application was approved in May of 2018 for $8.4 million in federal funding through the AAFC Canadian Agricultural Partnership Program and included about $3 million in industry contribution as well. We're currently in year 5 out of 5 for the cluster. So this is why we really wanted to do these cluster update webinars now. Since we've had 4 out of the 5 years completed for these sessions, we thought it was really valuable to provide industry with an update on how these projects are going so far. Here is a list of the cluster themes. We've broken them down into a few overarching categories. You can see listed here, science coordination. This is just the one that allows CGCN to administer the cluster and run everything. We have strategic management of grapevine virus diseases, cold hardiness and adaptation to climate change, sustainable management of soil, water, and crop quality. Obviously, the one we're here for today, optimizing the quality of Canadian wines. In order to increase and maintain market share of Canadian wines, both domestically and abroad, the Canadian wine sector needs to continually optimize the quality of the product while showcasing the environmental sustainability of the production cycle and fostering public trust. So the reason we're here today to talk about activities 12, 13, and 14, which look to address these issues with focus on the development of Indigenous yeasts for domestic wine production, field-to-winery management strategies for improving fermentation efficiency and tannin profiles in red wine, improved processes for sparkling wine production, and consistency in the marketplace. So that's just a bit of a teaser for the topics we'll be covering today. And then also as part of the cluster themes, we have crop protection and monitoring and knowledge and technology transfer, or KTT, which again is a CGCN responsibility. And part of that is this series of webinars that we're hosting today and throughout earlier in 2022 as well. With the exception of activity 1 and 23, which are run by CGCN, we've done cluster research updates now for all of these themes, if you count the one we're about to do today. So all of those past webinars are also available on our website and our YouTube channel if you're curious to learn more about the other themes as well. That's all I wanted to say about CGCN and the cluster. If you have any questions about the research that we contribute to, feel free to contact me anytime or check out our website. There's a lot more information there. Enough from me. Now we'll get to the exciting part of the day, the presentations. So I'll let Deb pull up her slides and I'll read through your introduction and then we can dive right in. So Dr. Debbie Inglis has been involved in the grape and wine industry for her entire life, as her family were grape growers in the Niagara Peninsula in Ontario, and her brother owned a winery. She herself is actually a second-generation grape grower. Debbie attained her PhD from McMaster University in biochemistry in 1995, and after a 3-year stint in the biotech industry, she joined Brock University in 1999 as an assistant professor in the Department of Biological Sciences, teaching in the Enology and Viticulture program. She took on the directorship of the Cool Climate Enology and Viticulture Institute, or COVI, in 2008, and now holds the title of full professor and continues to act as director of COVI. Debbie's research program first focused on issues of ice wine fermentation, but has expanded over the years to other red and white wine styles, along with biochemical issues in grapevine cold hardiness, and more recently, diagnostics for grapevine diseases. Without further ado, I will turn it over to Deb. Thank you.
Thank you, Ethan. Really appreciate the introduction. So today I'm here to talk about a program we've been developing for a few years, and I've been doing this in partnership with my colleague, Dr. Belinda Kemp. And it's called Tannin Alert. And we're looking to launch this new program to assess skin and seed tannin during grape maturation and at harvest to assist in winemaking decisions. So quite a few years ago, the industry came to us with a challenge that they were having in producing high quality red wines consistently year after year in a changing climate. In making red wine, you need skin contact for the color extraction. But the challenge is in extracting color and tannin from the skin, you want to do that without over extracting bitter tannins from the seeds. And this challenge becomes amplified in cool climate regions because the tannin development is a little bit different in cool climates than it is in warmer climates. So the fruit does not always reach optimal ripeness. And because of that, we need to be looking at not just bricks and acid development in the grapes, but also what we refer to as tannin ripeness. It's not really tannin ripeness, but it's the increase in skin tannin and the hardening of the seed coat that will reduce seed tannin from being extracted into the wine. So if skin color is not developed and skin tannin is too low, then winemakers have a challenge to try to really extract what is there from the skin, but at the same time without over extracting what's present in the seeds. So consumers are demanding a high quality, consistent product despite annual weather and vintage variation. The LCBO, which is one of our largest outlets for selling wine, wants red wines that are ripe, rich, and rounded. And that's short form for wines that are balanced in tannin. In some years, this requires greater intervention by the winemaker. And in Canada, we can be at a disadvantage in tannin development in our cool climate regions, just because of the seasons that we have. So what are these tannins that we're talking about? They're based on the structure of phenol, and the structure of phenol I've got here over on the side. They're found in various food products such as tea, dark chocolate, and of course grapes and in wine. And they're responsible for the astringent mouthfeel that we can get in wine. But tannins can also greatly impact the perceived quality of food products, such as the taste and also the color. And there are two main classes of tannins, the condensed tannins and the hydrolyzable tannins. And we're going to focus on the condensed tannins here. So the basic unit of condensed tannins is made up of one type of flavonoid known as the flavan-3-ol compounds. And this is a diagram of these compounds. I'm not going to be too heavy in the chemistry here, but this is a 3-ring structure. And you can have different substitutions at this R1, R2, and R3 position that can lead to all of these different variations of flavan-3-ols. These monomers are found in the skins and seeds, and catechin and epicatechin are really known for the main drivers of that bitterness in wine when they're over extracted, especially from the seeds. But those monomeric units will bind together to form dimers, like illustrated on the side here, or trimers or tetramers, or even larger complexes known as polymers. And these complexes, because of these hydroxyl groups, the OH groups on them, they're able to interact with proteins and polysaccharides that can make even larger complexes and affect the extraction during winemaking and even the taste of the final wine. So you know how sometimes when you taste wine and it's very astringent, very drying on the palate? It's because these chemical groups can interact with the proteins that are present in saliva in our mouth that are responsible for that lubricating effect. And if the tannins interact with those saliva proteins, they can actually pull them out of solution and they're no longer able to lubricate our mouth and we get that drying sensation. But tannins are also known to bind to pigments to create polymeric pigments. And this is really important for color stabilization. So I've just got a couple more slides with some chemistry here because I want to make sure everybody's clear on the importance of tannin in stabilizing red wine color. So if this is the anthocyanin pigment, that has red color to it, it can equilibrate into different forms depending on the pH of the solution. And one of those forms is actually colorless. And one of the forms will convert this red tone to more of a violet tone. And this is the quinoidal form. And this occurs as the pH starts to increase in the wine. But the majority of the pigment likes to reside in this colorless form. So if we don't have a way to trap the pigment in these colored forms, we end up losing color over time. And any winemakers in the crowd would be familiar with this if you're making a light colored rosé with very little tannin that's been extracted from the grapes. Over time, you tend to lose more and more color in that rosé because there's not enough tannin to trap it in a colored form. When we have tannin, such as, this is just showing catechin, the single monomer, but it could be catechin or a full tannin complex. If it binds to the pigment molecule, it traps it in this colored form, and it helps to stabilize that color over time. So tannins are really important to get into the matrix of the wine to help with that stabilization. So in the current winemaker toolkit, there are easy ways to measure Brix and get ideas of acidity, but there was no easy way for any kind of predictive analysis to look at the tannin level in the wine. And in speaking with the winemakers, they wanted something that would more realistically try to predict the challenges that they were going to see in making the wine to extract those tannins from the skin, but minimize from the seed. And so in speaking with my colleague, Belinda Kemp, who came on board as we were launching this program, she said, we really need to look at a way to separate the skin and seeds from each other and then do extractable tannin measurements on both the skin and seed and provide that information to the winemakers, because that will be much more informative to them than just grinding up the whole berries and giving them a full tannin extraction value that doesn't separate the skin and seed values. So we started on our journey of our Tannin Alert program, where we sample grapes from veraison to harvest. When we get them into the lab, we separate the seeds from the skins. We do tannin extractions on both the seeds and skins. And then we measure the amount of tannin extracted using the methylcellulose precipitation assay. So Tannin Alert is designed to be an easy to follow tannin measurement program for red grapes, and it will allow a grower or a winemaker to benchmark the skin and seed tannin in their sample against what's happening in the region. So we follow the tannin ripeness of red grapes, and we focused on 6 red varieties throughout this program, from veraison to harvest. We measure the skin and seed tannin separately for each variety, develop the Ontario Skin and Seed database for red grape varieties at harvest to benchmark the tannins, and that's the Tannin Alert. And then when a sample comes in, we can categorize the skin and seed tannin into low, medium, or high ranges based on the data that's in the database. And the idea here is to optimize red wine quality by accounting for tannin levels in the grapes at harvest and using that information to help direct winemaking options available for winemakers. So there were the 6 red varieties that we focused on. Initially were Cabernet Sauvignon, Cabernet Franc, and Merlot. And we then expanded into, and Pinot Noir, and then expanded into Merlot, Gamay, and Syrah. What we found over the years, and I'm going to share a little bit of this data, is that there are variety differences, site differences, within a variety, and vintage differences in the tannin profiles that we get from the fruit. And winemakers would need to measure the tannin in their grapes at harvest to document their profile prior to fermentation and compare their numbers to the database to then make management decisions. And it would be important to do this yearly because, again, we see such vintage variation and site variation, and of course, the variety variation as well. So what does this look like? So let me share with you some data that is looking at the extractable tannin on the y-axis and the date from veraison to harvest on the x-axis. And this is data from Cabernet Sauvignon from 2020. And this is from one of our sites where we would expect to see skin tannin increase from veraison to harvest and seed tannin to decrease from veraison to harvest as the seed coat gets thicker and prevents more extraction of the tannin as that seed coat hardens as we get closer to harvest and ripeness. And that's exactly what we saw in 2020 for Cabernet Sauvignon. So I thought I'd start with something easy. We saw a nice increase in skin tannin and a decrease in the seed tannin. But that didn't always happen. And this is a snapshot of data for Cabernet Sauvignon for 5 years. Each year from veraison to harvest is a different color. And this is all data collected from one of our sites. It's from site 1. And yes, we can see in some years, such as in 2016, we did see an increase in skin tannin happening here. But in other years, like in 2018, there was very little change in skin tannin during that period. Similarly, sometimes we see a drop in seed tannin, or sometimes we see very little change, or even a slight increase in extractable seed tannin. We also saw a variation between sites. So if we take a snapshot, again, this is for Cabernet Sauvignon in 2020, and compare the profiles from the 4 different sites, we see that for the skin tannin, the lines are not on top of each other. We've got variation from site to site. And that holds true for the values at harvest time. The seed tannins were a little more similar from site to site once we got a little further on in the season, but again, still some variability that we're seeing. So how would a winemaker use the Tannin Alert system to benchmark their tannin values against what is in the database? So when we collect all of this data from all the different sites through all the different vintages and put them into a database, and we've done this right now per variety, but we could collapse it all into all red varieties if we wanted to. We can do a distribution plot, and this is a histogram plot where we have the number of samples that test at a certain variety level on this axis, and we have the value of the tannin on the x-axis. And if we had a normal distribution, the histogram would look like this, where we have the majority of samples coming in at this medium value for tannin. We have a lower number of samples coming in at the low value and a lower number coming in at the high value. And we've separated this histogram to make these distinctions of low, medium, and high tannin based on the 33rd percentile and 66th percentile of the distribution. So when we look at a variety like Cabernet Sauvignon, and we take a look at the skin and the seed values in the database, now with all the years that we've been collecting data, and this actually goes back to 2015 because we started this program before the cluster, we can see that we have a range for low, medium, and high values for skin and seed tannin for Cabernet Sauvignon. Then we would get a sample that would come into the lab. We would measure the skin and seed tannin and report that against the database. So here the sample had a medium range of skin tannin and a high range of C tannin. This is a difficult scenario to work with in winemaking because you don't want to over extract those C tannins, but you're trying to get all the skin tannin out of the grape. For Pinot Noir, it's a different distribution. It's a different variety. It has a different distribution. And when a sample came into the lab in this particular year, we found that the skin tannin was high. This was actually from 2020, and the C tannin was low. So this is a better profile for winemakers in order to minimize the C tannin extraction while they're trying to get that skin tannin out. So we carried out some winemaking projects using this system, and I'm going to walk through a couple of the projects that we did with Pinot Noir so you can see how we use the system. So in 2019, we had control grapes where we just did routine red wine production. We had one treatment where we added some skin tannin that was commercially available into the ferment. We also tried a pectolytic enzyme that would help break down the skin to extract skin tannin. And then we did a combination of skin tannin addition and the enzyme. We had some really promising results from the pectolytic enzyme treatment. So the following year, we further expanded that research in looking at two different types of pectolytic enzymes on their own and in combination. So when we got the grapes in that year, you can see in 2019, for this Pinot Noir, we had a medium range of skin tannin and a high range of C tannin. Whereas in 2020, we had high skin tannin, low C tannin. This is the more desirable profile that a winemaker would want. This profile, we have to be really careful about not over extracting the C tannin through our winemaking processes. Otherwise, the wines are going to become too bitter and astringent. So from the 2019 treatments, we saw immediately after pressing the wines out, the skin tannin really didn't have much of an impact on improving the tannins in the wine, but the enzyme treatment did increase the tannins in the wine. And that relationship, and the combination saw the same as just the enzyme addition. And that profile continued 3 months post-press and also 9 months post-press. So there is an increase in extractable tannin when enzymes are added in comparison to the control, but the skin tannin addition didn't show a difference. So the following year, we decided to try the two different enzyme treatments. And again, both enzyme treatments alone increased the tannin, and in combination, we didn't see an additive effect, but we also saw that increase over what we saw with the control. That relationship held at 3 months, and it also held at 6 months. So at each time point tested, the addition of the enzymes, whether they were pectolytic or cellulosic, increased the concentration of extractable tannins in the wines when compared to the control. And this is just a different way to view that same data. But in this profile, we're actually able to see for each treatment what the tannin stability was like over time. And again, with the control treatment, we're losing tannin over time as these wines age. Whereas when we had both enzyme treatments used, we had better stability of the tannin over time. So over the years, there have been many different winemaking trials that have been carried out by Belinda and grad students and fourth-year students working with us on this project, looking at Pinot Noir, various commercial skin and seed tannin preparations. For Cabernet Franc, looking at pre-fermentation skin disruptions or post-fermentation pressing pressures. Cabernet Sauvignon, we looked at extended maceration time or pre-fermentation skin disruption, enzyme addition, and Sangnier treatments. This was, and sorry, the students carrying out the work are listed here for each of these projects. We also looked at skin and seed tannin addition in Gamay and the inclusion of whole bunches. Just last year, Belinda started looking at flash détente treatments for Merlot, and also the use of liquid tannin post-racking and post-filtration for Syrah. So we hope to launch, our plan is to launch Tannin Alert as a service this summer, where we did a testing last year in 2021 with a couple of winemakers, just to make sure that we could get the right turnaround time and the information back to them in a timely manner. And we've got this down to about a 3-day turnaround time from sample drop-off to when we can get the test results back to the winemaker. And the results will provide not only the skin and seed tannin value, but also benchmark it against the database. So you'll know whether you're in the low, medium, or high range. And it will provide access to the Tannin Alert website, so you can look at various treatments that might be applicable to you. And we hope to target this at about $100 per sample based on the amount of time it's going to take us and equipment usage to get those values going forward. So from this project, we've been able to create an online tool for grape growers and winemakers to improve red wine quality. The industry, actually, when we were first pitching this project to one of the funding agencies, felt that using Tannin Alert, we'd be able to increase domestic market share by 21% in 10 years and increase 800 new jobs, and also increase the average price point of domestic wines. So these would all be really positive things for the industry going forward, and certainly significantly increase the reputation and branding of Ontario wines right now, because the database is just based on Ontario grapes, but this could be expanded nationally. So just in my last couple of slides, we've had a lot of partners over the years. So I'd like to thank Ontario Grape and Wine Research Inc., Alan Schmidt, Dave Shepard, Emily Bourrier, and Craig McDonald for being partners initially when we developed this program. All of our grape donations that have come from growers and wineries over the years for all of those 6 red grape varieties and various sites. My collaborator and co-PI on this project, Dr. Belinda Kemp, grad students Leah DeFelice Renton, Charlene Marcotte, Robin Holford as a 4th-year thesis student, and all of our technical support in the Institute. And of course, our funding partners, which includes CGCN and Agriculture and Agri-Food Canada, along with NSERC and OCE and OGWRI that helped us start this project back in 2015. And just as a very last slide, I would just invite all that are listening here today to the International Cool Climate Wine Symposium that we're holding at Brock this July 17th to the 21st. And you can hear so much more about challenges and solutions to issues we face in cool climate grape and wine production in Canada. Thank you.
Perfect. There we go. Sorry, I had a bit of technical difficulty myself. Thank you so much, Deb. That was wonderful. And I would echo the sentiment about making sure to come to ICCWS. CGCN will actually have a booth at the trade show. So if you want to come say hi to me and learn about a lot more fascinating projects, that's a great way to do so. Thanks, Deb. All right, we'll move on to our next speaker here. So Gavin, if you want to go ahead and get your slides set up, I can read your introduction for everybody. And just as a reminder to our attendees, again, if you do have questions at any point, you can type them into the Q&A button at the bottom of the Zoom screen, and we'll address them at the end of all the presentations. So Dr. Gavin Kernaghan obtained his BSc and MSc from the University of British Columbia before completing his PhD in the Department of Biological Sciences at the University of Alberta. From there, he held several postdoctoral fellow positions across the country at the University of Alberta, L'Université de Québec à Montréal, and Southern Crop Protection and Food Research Center at AAFC in London, Ontario. Since 2006, he has served as a professor in the biology department at Mount St. Vincent University in Halifax, Nova Scotia. His research focuses on the ecology of fungi, particularly the biotic and abiotic factors that govern the structure of fungal communities, with a focus on symbiotic relationships. I will turn it over now to Dr. Kernaghan. Thanks for being here.
Thank you very much. Title of my talk today is Improving Wine Quality Through Mixed and Sequential Fermentations with Indigenous Yeast. And this is part one, assessing vineyard yeast communities. So most commercial wine fermentation involves inoculation with Saccharomyces cerevisiae, which is our classic wine yeast, and it gives us quite a predictable fermentation. Saccharomyces cerevisiae also tends to dominate spontaneous fermentation. So even when we don't inoculate with yeast, we still get Saccharomyces cerevisiae dominating, and that's due to its alcohol tolerance. It can handle a lot of alcohol, and it outcompetes the other yeasts in the system. However, early in the fermentation, we have other yeasts, native yeasts, or what we call indigenous yeasts, which are on the grape skin and can produce a range of compounds that influence wine quality. And this is part of what we call the microbial terroir. These indigenous yeasts can make alcohols such as glycerol, they can make sulfur compounds, they can make phenolic compounds, and hundreds of volatile metabolites which can influence grape wine quality. So the objectives of this project were to find non-conventional vineyard yeasts to use in combination with Saccharomyces cerevisiae, either in mixed or sequential fermentations to improve wine quality. When I say mixed fermentation, I mean to use an indigenous yeast and Saccharomyces cerevisiae at the same time. When I say sequential fermentation, I mean to start with the indigenous yeast, start the fermentation with indigenous yeast, and finish the fermentation with Saccharomyces cerevisiae. This work falls into three parts. Part one is what I'm talking about now, the assessment of vineyard yeast communities. So essentially, we need to know what's in the vineyards so that we can work with them. Part two is the physiological characterization, or finding out the fermentative properties of the yeasts. And that will be Lihua Fan in the next talk up. And part three is actually making wine, fermenting with these indigenous yeasts. And that's some ongoing work. So Nova Scotia has a burgeoning cool climate wine industry. We now have 16 commercial wineries in the Annapolis Valley region. We worked with 8 of these wineries, 5 with conventional viticultural practices. These are the ones with the red dots, and 3 with organic viticultural practices. Those are the yellow dots. So sample collection and processing. We collected Lacadie Blanc grapes. They were collected at harvest, and we have 2 years of sampling from each vineyard. The grapes were crushed sterilely and split into 4 replicates per sample. And we collected fungal DNA, which includes the yeast DNA. We sampled that from initial must before fermentation, and again after spontaneous fermentation was allowed to complete. The relative abundance of the fungal and yeast species in each sample was then identified using 2 different next generation DNA sequencing platforms. We used the Illumina MySeq platform, and we also used the PacBio Sequel 2 platform. Now, the Illumina MySeq platform gives hundreds of thousands of short DNA sequences per sample, so it's very good at detecting rare species. On the other hand, the PacBio Sequel 2 only gives thousands of sequences, not hundreds of thousands of sequences, but each sequence is much longer, and that gives better species identification. So we use the data from each platform to complement the data from the other platform to give a more complete picture of the vineyard yeast communities. So as far as data processing or bioinformatics, this project is very bioinformatic heavy, but I won't dwell on it. I'll just tell you that we used FastQC, was a software for sequencing sequence quality assessment. So we use that to kick out bad sequences. The sequences were then processed and identified by comparing our next generation sequences to existing sequences on the GenBank database. And this was done with software called the SEED2 pipeline. So these are screen captures from SEED2. The one on the left is just showing lining the sequences up and comparing them. And the one on the right is just the top of the list of the output. This would actually be quite a long list. And we can see from this that the most common sequence type, most common yeast found in this particular sample was Oreobasidium pullulans, which is a black yeast, very common on grape leaves. And we found that there were 22,089 of those sequences. So it's a lot of data. In some cases, we also confirmed the yeast identification by what we call manual phylogenetic binning. And we did this for tricky groups like Saccharomyces, for instance. There are several species of Saccharomyces, and they can be difficult to tell apart with our short next generation sequences. So what we did was to build a phylogenetic tree or a family tree based on reference sequences from GenBank, and then we would add our unknown sequences to that. So here in bold, we can see here's a sequence from Vineyard number 7, which clearly falls into the Saccharomyces uverum group. We've got another one down here in bold from Vineyard number 4, which clearly falls into the Saccharomyces cerevisiae group. So basically, this was a way of confirming our identifications for tricky groups. So as far as results go, I'm going to start just by showing you results from one example vineyard, and this happens to be an organic vineyard. In this diagram, the 2 years of sampling are pooled, and the 4 replicates from each sample are pooled. And we're only showing you species which were greater than 0.1% of the total. And in this figure, it's broken down in between the things that we found in must and the things that we found in the resulting wine. So the first thing to note is that the must has more yeast species. There were 159 yeast species in the must in this sample and only 72 in the wine. The other thing we can see is that almost all of the non-yeast fungi were in the must. So here in red, these are the non-yeast, the filamentous fungi, the molds, etc., the things that are not actually yeast. So those are almost all in the yeast, and this skinny red line here is showing you that there is just some small carryover into the wine, but those are mostly in the must. The other thing we notice is that the must is dominated by non-fermentative yeasts. So these are things like this Oreobasidium pullulans here, and Phylobasidium, several species of Phylobasidium, and down here in green we have Vishniocozyma. These are what we call basidiomycete yeasts. They are yeast, but they're not fermenting alcohol. I'll come back to those soon. So anyway, those are dominating the must. When we get into the wine, it's a very different situation. Once that spontaneous fermentation has happened, almost all of these non-fermenting basidiomycete yeasts go away, and we're left with a fair amount of our classic wine yeast, Saccharomyces cerevisiae, but we're seeing an even larger amount of Saccharomyces uverum. This is a fermentative yeast, so it's making ethanol, and it's essentially the wild cousin of the Saccharomyces cerevisiae. And I'll talk more about that later as well. So this is the same results from the same vineyard, the same example organic vineyard. I'm just going to break it down a bit. This is broken down into 4 replicates. And on the left, we have the pre-fermentation or the must data. And on the right, we have the post-fermentation or the wine data. And across the top 2 graphs are 2018 samples, and the bottom 2 graphs are 2019 samples. In the must in 2018, we see is dominated by a few non-fermentative yeasts, but then when the spontaneous fermentation happens, it becomes dominated by Saccharomyces uverum, which I said was the wild cousin or the indigenous cousin of Saccharomyces cerevisiae. So it's completely dominating the 2018 wine. The 2019 situation was slightly different. The must was still dominated by non-fermentative basidiomycete yeast, but we're getting a higher diversity. We're seeing more of them now. But then in the wine from 2019 is quite interesting. We're getting 50% of the replicates are dominated by Saccharomyces cerevisiae, and 50% are dominated by Saccharomyces uverum. Even though these 4 replicates were taken from the same sample of grapes, the same clusters of grapes, we're getting sort of a random flip-flop of either Saccharomyces cerevisiae or Saccharomyces uverum. Saccharomyces uverum is a non-conventional fermentative yeast, so it does make ethanol, and it's very common in cool climate vineyards, so we expect to see it all across Canada. Wines made from different strains of Saccharomyces uverum quite variable in flavor. We've done this. We've made wines from different strains of Viverum, and they really are quite different. There is one strain that's been previously commercialized, not by us, but there is one on the market, so people know about it. The fermentation by Saccharomyces viverum is not terribly strong. It doesn't make a lot of alcohol, so it's leaving some residual sugars. And it seems that this might be a good possible candidate for sequential fermentation with Saccharomyces cerevisiae. So if we had a valuable Saccharomyces viverum isolate, we could ferment with that first, and then finish the fermentation with Saccharomyces cerevisiae, just to make sure that the sugars are all used up and enough alcohol is made. So that so far has just been data on one individual vineyard. I'd like to expand now and talk about comparisons across all the vineyards we looked at. In general, overall, yeast communities were more diverse in the organic vineyards than they were in the vineyards with conventional viticultural practices. And not only that, but the species makeup of the yeast was different between the two viticultural practices. And we can see that in this diagram. This is an ordination, and for those of you not familiar with ordinations, points that are closer together will have more similar yeast communities, and points that are further apart will have more dissimilar yeast communities. And so in the ordination, we see that the conventional vineyards here with the open circles are all relatively similar, and the organic vineyards with the closed circles are distinct and relatively similar to each other. So these community differences between the conventional and the organic vineyards seem to be attributable to these non-fermentative basidiomycete yeasts that I was describing earlier. And that's possibly due to their sensitivity to the synthetic fungicides used in the conventional vineyards, and perhaps the fermentative yeasts like Saccharomyces that are more common in the conventional vineyards are more resistant. We can see more information on the importance of these basidiomycete yeasts in this data set. This is indicator yeast species. So it's an indicator species analysis for organic and conventional vineyards. So here with the organic cultivation practice, we get a long list. This is 13 different yeast species, which are statistically associated or characteristic of organic vineyards. Whereas down at the bottom, for conventional, we only see 3 yeast species which are characteristic, statistically associated with the conventional vineyards. Not only that, all 16 of these are the basidiomycete yeast, the non-fermentative basidiomycete yeast. So it looks like they are very important in driving this difference between the organic and the conventional vineyards. So a bit more on non-fermentative, what we call basidiomycete vineyard yeasts. These have been relatively ignored until recently, but they may end up being more diverse. We think they're more diverse than the fermentative yeasts in vineyards. Some of them produce quite a few volatile compounds, resulting in a range of aromas like cocoa, almond, whiskey, malt, banana, nutty flavors, pineapple flavors, rose, and honey. They have enzymatic activities such as pectinase and esterase, and these may increase juice yield from the grape and may also affect flavor and aroma. The non-fermentative yeasts can also produce glycoproteins that can enhance wine aroma. And although these yeasts are not directly involved in ethanol production, they may still impact wine quality. So in conclusion, this is really the first comprehensive assessment of vineyard yeast in Atlantic Canada. We found a high diversity of both fermentative and non-fermentative yeasts in both musts and spontaneous wines. We found that Saccharomyces viverum can dominate our spontaneous fermentations, and they may be a good candidate for sequential fermentation, along with Saccharomyces cerevisiae. And the non-fermentative or basidiomycete yeasts tend to be more abundant in the organic vineyards and may be a valuable resource for improving wine quality through co-fermentation with Saccharomyces. And with that, I'd like to thank CGCN, Grape Growers Association of Nova Scotia, Agriculture and Agri-Food Canada, St. Mary's University, and Mount St. Vincent University. Thank you.
Perfect. Thanks so much, Gavin. It was very informative and transitions perfectly into the next presentation we're about to see. Of course, you spoke on part one of the project. Now I'll invite Lihua to go ahead and pull up her presentation slides while I introduce her, and we can move on to part two of this discussion. Dr. Lihua Fan is a food microbiologist working at the AAFC Kentville Research and Development Center in Nova Scotia. She joined AAFC in 1998 and has successfully led or participated in many national and international collaborative research projects. Her research has been focused on food safety and quality, including developing novel natural antimicrobials from plant and bacterial origins for food protection, controlling heat-resistant molds to extend shelf life of processed food products, developing innovative technologies to control post-harvest decay of horticultural produce, ensuring microbial safety and quality of whole and fresh cut fruit and vegetables, and applying fermentation technology to improve flavor and nutrition of value-added foods. Dr. Fan is a professional member of the International Association of Food Protection and the Institute of Food Technologists. She is a certified food scientist and an adjunct professor at Dalhousie University and St. Mary's University in Halifax, Nova Scotia. Dr. Fan, thank you for being here, and I'll turn the mic over to you.
Thank you for, can you hear me?
Yep, we can hear you just fine. Yeah, thanks.
Okay, thank you for this opportunity. The presentation title is Improving Wine Quality Through Mixed and Sequential Fermentation with Indigenous Yeast. Gavin has already talked about part one. I'm going to focus on part two, fermentative properties of vineyard yeast. Why are indigenous yeasts important? The natural microflora of the grapes of the viticulture region significantly contributes to the unique quality characteristic of wine. Indigenous yeast can influence the alcohol content, pH, viscosity, color, the concentration of sulfur compounds, phenolic compound, and the volatile profile, contributing to the flavor and the aroma of unique wine quality. So the objective of our research project, the overall goal is explore the potential of Nova Scotia indigenous yeast to improve wine quality through nature, mixed, or sequential fermentations with commercial yeast starters. Here we talked about Saccharomyces cerevisiae. There are a few specific objectives in our research. Number one, isolate, purify, and identify indigenous yeast from a vineyard in Nova Scotia. Number two, investigate the fermentative properties of indigenous yeast and select promising yeast strains for wine production. Number three, conduct natural fermentation trials using grape mast, determine microbial changes, microbial quality, microbial population during fermentation, and the chemical composition of the wine products. Number four, relate the indigenous yeast population, microbial communities in vineyard, and the fermenting mast to the quality of the resulting wines. So the first step is isolation and purification of yeast. We collected grapes from multiple vineyards in Nova Scotia and isolate yeast from grape and fermented mast using yeast selective medium. And then we purify the yeast strains for further use. For these purified yeast strains, we will do the molecular identification. This includes extract DNA from yeast cells, amplify selective loci, sequencing DNA, editing chromatograms, analysis gene sequence data, and to determine the yeast species. After identification, we have conducted many different characterization tests. This includes the ethanol tolerance test. Yeast ethanol tolerance was tested at a different concentration from 0% to 20% ethanol levels using a bioscreen C equipment. The optical density OD value was recorded for 48 hours. Here we can see the picture of bioscreen C instrument, and this is the microplater we used for inoculate different samples. After identification, for this week, we can see the different growth curves generated by the bioscreen C equipment. Usually within 48 hours, we can generate more than up to 200 different growth curves. So yeast surviving the alcohol concentration were determined based on these growth curves, include the lag stage and the growth slope and the maximum OD values here. So these growth curves, we can see that is under different concentration of ethanol percent. With that, we do the statistical analysis and determine which concentration and can survival for these yeast strains. For sulfur dioxide tolerance test, yeast strains were inoculated into test tubes containing YPD broth and a different concentration of sulfur dioxide. We included 0% up to 60 milligram per liter. The samples were incubated at 25 degrees for 72 hours, and the OD value was measured using a spectrophotometer. The bottom one is for the beta-glucositis activity test. Yeast strains were streaked onto agar plate containing YNB, that is yeast nitrogen base, aptone plus ferric ammonia citrate, and they incubated at 25 degrees for 8 days, where commercial yeast starter EC1118 was used as a control. From this Petri dish, we can see the beta-glucositis activity was indicated by the dark brown coloring in the medium. So we can decide if that is beta-glucositis positive or negative. So we compare that with controls, commercial start culture. This is for the hydrogen sulfide production test. Yeast sample were plated on biggie agar using the spiral plater method and incubated at 25 degrees for 72 hours. The production of hydrogen sulfide was indicated by a browning color in the colonies, which became darker with increased hydrogen sulfide production. A 5-point scale ranging from 1 to 5 to reflect white to dark brown color was used to evaluate the hydrogen sulfide production. This one is a very white color, and then this is a little bit browning color. The bottom picture is with very dark browning color. We are very excited to select some yeast strains for our wine fermentation. So far, we have more than 300 yeast strains were identified and characterized for their fermentative properties. And promising yeast were also selected based on the ethanol tolerance level higher than 6%, sulfur dioxide tolerance level higher than 20 milligram per liter, lower hydrogen sulfide production, beta-glucositis activity positive, and other enzyme tests. So selected yeast strains include Saccharomyces yeast, such as Saccharomyces cerevisiae, Saccharomyces ovarium, and the non-Saccharomyces yeast such as Hansania spora, Pichia, Xerotorola spora, etc. To further study Saccharomyces yeast and non-Saccharomyces yeast and the microbial communities, we conducted natural fermentations. We use grape cultivar of Lacadie and Pinot Noir. We selected the vineyard, 8 Nova Scotia vineyard. 5 of them are conventional vineyard, and 3 of them are organic practice vineyard. For this research work, we have done 2 years research for all of these natural fermentation sample. We collected 2 sampling years per vineyard. Here we can see the picture showed we use a stomach blender to press grape and get the mast for lab scale fermentation. During the fermentation and after the fermentation, we evaluate Brix, pH, titratable acidity, weight loss, fermentation rate, volatile acidity, microbial changes, microbial populations, and the chemical compositions. This table is an example of microbial evaluation during fermentation from day 0 to day 28. At a 3 to 4 days interval, we took samples to evaluate total yeast population using YPD agar. We evaluate non-Saccharomyces yeast population using Lyson medium. We evaluate total plate count using TSA medium. Finally, we also evaluated lactic acid bacterial population using MRS agar. For chemical analysis, we measured ethanol, glycerol, sugars, organic acids, phenolic compounds, and volatile compounds using HPLC and GCMS. This table is an example showing sugars and organic acid of natural fermentation product. The left column here includes grape mast processed from different vineyards in Nova Scotia. The value number in blue color, the top one, indicates the result before fermentation. The value number in red color are result after fermentation. We can see that after the fermentation, for some samples, the ethanol concentration can reach to 10 to 11%. Residual sugars was close to 0, where the other samples cannot finish fermentation with lower ethanol concentration and higher residual sugars in the final product. In addition, the concentration of glycerols and the malic acid, which contribute to sensory quality, are very different among our final fermented product. We also measured the phenolic compounds and compared the one sample fermented use our own selected yeast strains and those fermented with the commercial yeast. So we compared the overall phenolic compounds profiles. To further determine the specific phenolic compounds, we also measured these compounds at different wavelengths for our samples. Also, we compared that with the commercial yeast samples, yeast fermented samples. So we try to decide what is the compound in our samples, what are they different with the commercial fermented samples. So based on our studies on fermentative properties of indigenous yeast and natural fermentation trials, this slide covers our research findings. More than 300 indigenous yeast strains from grapes and mast were isolated, purified, identified, and stored at -80 for our long-term research use. Characterization of yeast strains revealed a very wide biodiversity within each genus species. Some natural fermentations progress efficiently, where the others were very slow. Non-Saccharomyces yeast were active at early fermentation stage, indicating that mixed or sequential fermentation trials are necessary. Chemical analysis showed that residual sugars, ethanol, glycerol, some phenolic compounds, and volatiles are different in natural fermented wine product, suggesting that yeast species and yeast communities played an important role in wine quality. Our research result will help create a yeast bank database and provide useful information for the grape wine industry with more yeast wine specific data that can be utilized for marketing and promoting different texture and aroma profiles in their final wine products. I would like to acknowledge Canadian Agriculture Partnership, Agroscience Program, AAFC, Canadian Grape Wine Certification Network, CGCN, the Grape Grower Association of Nova Scotia, GAN, Nova Scotia Grape Growers, and our research technician at AFC for their support for this project. With that, thank you very much for your attention.
Perfect. Thank you so much, Lihua, for sharing your expertise and for tying together part two when Gavin spoke about part one of your work. Thank you very much. And for those of you attending, you saw as well that Dr. Fan had her email in the last slide there. So if you have any follow-up questions, you're welcome to use that to reach out, no doubt. Last but not least, we have Dr. Belinda Kemp here to present on activity 14. So I'll give her the opportunity to pull up her slides. And I will introduce her right now. Dr. Belinda Kemp is currently the Senior Staff Scientist in Enology at Covey at Brock University here in Ontario, and an Adjunct Professor in the Faculty of Biological Sciences. Belinda gained her PhD in 2010 at Lincoln University in New Zealand, studying Pinot Noir, tannin, flavor, and sensory characteristics of wine. As well as a scientist, Belinda has gained practical winemaking experience in commercial wineries in New Zealand and the UK. Her research focused priority areas of the Canadian grape and wine industry for premium wine production, including red wine tannins, optimum conditions for Maillard reaction in sparkling wine, influence of yeast nutrients on sparkling wine flavor, off flavors in wines, and the impact of sugar and dosage solutions on sparkling wine flavor and aging. She currently organizes Fizz Club for Ontario sparkling winemakers to disseminate research results and serves on the Vintners Quality Alliance of Ontario, or VQAO, Wine Standards Development Committee, and the VQAO Sparkling Wine Regulations Committee. Belinda is thrilled to be presenting the Great Chardot Swap for wines at ICCWS 2022, the culmination of 3 years of collaboration with 12 different Niagara wineries. Belinda, thank you for being here, and I'll turn it over to you.
Thank you, Ethan. Can you hear me okay? Yeah. Okay, thank you. Thank you for inviting me to talk today. And yes, I just want to, another reminder of the Core Climate Wine Symposium there right on front of the first slide. So this part of the CGCN, which Debbie, who you heard speak earlier, my colleague Debbie is a co-PI on, is about looking at improving sparkling and still wine quality, particularly looking at volatile acidity, but in relation to acetic acid and ethyl acetate, and looking at some strange honey dusty off flavors that we encountered some time ago, and particularly focused is sour rot and the effect that sour rot has. The more sour rot you have in a fermentation, the effect on the final aroma compounds or compounds in general. And one of the ways that we tackled this was with a Canadian Indigenous yeast that my colleague Debbie's been involved in. It's called CN1, and it has actually, it is actually a Saccharomyces euvarum yeast, like Gavin, whether it's the same one, I don't know, it's beyond my abilities, but certainly it's a Saccharomyces euvarum yeast, and Debbie is the expert on that if anybody has any questions about that later on after this presentation. So I'm just going to cover a few things today because so much data over the years of the winemaking for this because of doing sparkling and the Pinot Noir red wine. So I'm going to give you a little bit of a background. I'm going to mention what these two honey compounds are. I call them honey compounds. It makes them sound really nice, and they're not. When they're combined together, they're sort of dusty, musty, like my grandmother's wardrobe used to be, sort of, but with a tinge of honey. Not sure that's particularly helpful, but that's the kind of smell and taste you get. I'm going to talk about the experimental design, just actually what we did. And we're going to look at the acetic acid and ethyl acetate levels in Pinot Noir red wine in 2020, because that was the year in the red wine Pinots that we had sour rot, whereas in 2019, when we did it, we had botrytis. And then finally, the base wine and sparkling wines, I'm just going to show you some information about these two musty honey compounds once I've explained what they are. So we'll have a look now. So just a reminder, I know the growers and winemakers are quite familiar with this, but just a reminder of what to look out for grape rot. These photographs are courtesy of our colleague Wendy over at Amafra. And on her site, you can have a look and learn a bit more about sour rot. I'm not going to go into too much detail, but just briefly. Sour rot's caused by a combination of physical factors. So basically that means like berry splitting, maybe from wasps or birds, microbial factors, including yeast, bacteria and fungi. And of course, it can be spread by the lovely vinegar flies. You can smell it. When it gets to a certain level, you can smell it at the end of the row. Non-Saccharomyces yeast are involved in the rotting process, but these yeasts differ depending on which region. Whether they differ by site, I don't know, but possibly the answer to that would be yes, because of looking at the data that Gavin produced earlier for Nova Scotia. Sour rot does decrease Brix levels because it converts the sugar into nasty compounds like acetic acid, gluconic acid, uronic acid, ethyl acetate, and of course glycerol. But the most important for us is the oxidation of ethanol into acetic acid. And here's the link for you to have a look at the Ontario Grape and Wine Research page to have a look a bit more about some of the projects that our colleague Wendy has been involved in and tells you a bit more about sour rot. Okay, one of the important things for us with these compounds in sour rot is the detection levels. At what point is too much? And the detection level is actually the concentration at which 75% of panelists can correctly identify or correctly detect the compound. So for acetic acid, it's 0.8 grams per litre, and the maximum permitted in Ontario is 1.3 grams per litre of acetic acid. And I think part of that is due to wine style. The ice wine tends to have a little bit higher. The ethyl acetate, however, for white still wine, it's 170 milligrams per litre, and red still wine, 160. Now, these become a bit more important a bit later on when I show you some of the data and information. So these two honey dusty grandmother's wardrobe smells I was trying to sort of explain earlier, let's just tell you a little bit about those. One is phenol acetic acid. It's a monocarboxylic acid, and it's actually the precursor to its much nicer friend, not much nicer compound, which is ethyl phenol acetate, which is much nicer compound. However, phenol acetic acid is a really interesting compound. It's a toxin, it's a plant metabolite, it's also a Saccharomyces cerevisiae metabolite, which is interesting. And again, you'll see in a moment. It's also a plant growth retardant and even an allergen and an auxin. So it was difficult to get into the country. You have to get permission to buy it and how to store it and look after it because of all these things that it can be involved in. It's also it comes from the degradation of phenylalanine, the amino acid, and they think at the moment that happens through the shikimic pathway, acid pathway, but we don't know too much about that at the moment, or at least certainly to the best of my knowledge, we don't know that. But certainly a really interesting compound to look into further. with regards to vines, vine leaves, and grapes and wine. Ethyl phenyl acetate is a much nicer honey compound, but the thing is with these compounds is they're highly unstable. So they can change during aging. And of course, because of phenylalanine and phenylacetic acid sort of coming directly from it, one of the other things is it will do things like increase, that this compound will increase if you use purely DAP, for instance, for adding for first or second fermentation in a sparkling. But there's other things. It naturally increases during aging on lees, for instance, because it's being produced coming from the yeast lees and any leftover nitrogen left over in the fermentation. So unstable, and sometimes 1 month you can taste it during aging of sparkling base wines, sparkling wines, and then 6 months later it's not there. And part of that is because of this aspect of the changes and its instability over time. So these two have detection thresholds. For phenylacetic acid in red wine, it's only 73 micrograms per litre that you can actually detect it at. And it's quite, I mean, we're so used to smelling it in our lab that we can detect it quite swiftly, but it is this dusty, musty, mixed with sweet honey smell. The ethyl phenyl acetate is much higher. You can detect it at 263 micrograms per litre. Then there were studies on red wine to determine at which point is too high. And the interesting thing here is it's not one compound or the other compound, but when they're present together at levels where the ethyl phenyl acetate is over 140 micrograms per litre, and the phenylacetic acid is over 700 micrograms per litre, then from then on is too much. But it has to be, they're the levels together. And we tried determining whether that was also the case in sparkling wine, but unfortunately we couldn't be sure because we didn't have the 75% agreement that we needed. So this again becomes important a bit later. So our project aimed, with that background information, was to actually, first we wanted to quantify these compounds associated with sour rot in both clean and infected Pinot Noir grapes during ripening, which we've done. We also wanted to aim to produce a Pinot sparkling wine and still red wines with varying amounts of sour rot. And again, it was to compare or determine the concentrations of these compounds. But the last really important part of this is, now that we've done those two parts, was to test the Brock yeast CN1, which, just as to remind you, is a Saccharomyces urarum, because it's already been shown to reduce acetic acid in wine. So we wanted to trial it for first fermentation on sparkling and for the Pinot Noir still wines, where there are different amounts of sour rot in them, to see if it reduces again acetic acid and ethyl acetate and these honey compounds. So you can see there's quite a lot there. Now the first thing was the sparkling wine design. This is quite different from the red wine. The sour rot bunches, we all went out and picked an awful lot of sour rot bunches, and then we whole bunch pressed them, cleaned up, and then we whole bunch pressed the clean bunches, kept them all separate, and then we actually added different percents in litres of the juice from the whole bunch pressing to the clean fruit, and that's how we came in litres to get those percentages. We did as we normally do with sparkling wine, racked it, and we inoculated we had triplicate replicates. And we inoculated with EC1118 and the CN1 yeast just for first fermentation. Second was EC1118 only. So we ended up with 30 base wines and sparkling wines. The next thing was the analysis. So for juice, we did malic, Brix, acetic, the standard lab analysis, but we also added in the two honey compounds. For the base wines and the sparklings, we also did that, including the residual sugar, the alcohol, the two honey compounds, that we were looking at as well. So then the red wine's different because here we had to do it by rate, the percent by rate. So again, we handpicked the sour rotten berries and got a team together who were thrilled to be able to spend the morning pulling off individual berries off bunches. And we crushed and destemmed the clean bunches, and then we added in kilograms to make up the percentage. So 20% of a 25 kilogram ferment would have been sour rotten berries. So we did a normal red wine method for covey, which is punch downs twice a day and fermentation monitoring till dryness. We did MLF and racked and bottled. Here, the lab analysis differs for the white wine because we saw the botrytis in 2019 instead of the huge amount of sour what we were really hoping for. And so we included in the lab analysis gluconic acid and glycerol, as well as the other lab analysis like we did. But again, that was done on juice, on base wines and on the finished sparkling wines. So let me just show you and start off by showing you the acetic acid levels in the Pinot Noir juice in 2020. So this is a juice which had more sour rot in. And the top picture to look at, A, is the acetic acid levels in Pinot Noir red juice. And you can see on the left there, the acetic acid in CN1. So this is before inoculation. This is just the starting acetic acid. And in the green, the acetic acid levels before we inoculated in EC111. So pretty similar between the ferments there. What we see in the second graph at the bottom there in B is now we see the acetic acid levels in the Pinot Noir wines. And on the left again in the blue is the CN1 yeast. So you can see at the higher level of 40%, we still have higher acetic acid, but not much. It's actually, if you look at the scale, it's 0.38 on that, which is the mean of that one. But you'll see that CN1 20% actually ends up being a similar, or I'll say the same concentration in the wine compared to none at all, which is an interesting aspect of CN1. But you'll see here as well, what's really important is the green on the right of, on my right of graph B are all the same. No matter how much sour rot was in there, they still end up very similar, and of course are not statistically significant at all. So that's quite interesting. You can still see that there is, again, CN1 showing that decrease in acetic acid. What we see is a slightly bigger difference in the ethyl acetate in the same wines. Again, CN1 yeast, the Uvarum yeast, is on your left, and you can see that, we tested it in juice just in case there was no ethyl acetate in the juice. But what you see here now is you see after fermentation, the CN1 yeast has much, I'll say much, I have to be careful with that word, but has lower ethyl acetate concentrations than the EC118 on your right in green. But we also found a statistical significance between the two yeasts. But the only difference between the treatments was between the three EC118 ones, which is what those letters are on the top. So that's the ethyl acetate. Bring you back to remind you of the levels of the two honey compounds. So we're looking at 73 micrograms per litre of phenol acetate, acetic acid, and ethyl phenol acetate 263. So let's just have a look at what we've got here. So here for the base wines, after all the addition of the different percentages of sour rot juice, we see stepping stones up in both compounds. Phenol acetic acid is much higher, you can see there in the base wines, than its corresponding nicer compound, the ethyl phenol acetate. So that's what happened with fermentation, our first fermentation. Now, the second fermentation happened with EC118. So let's just take a look at this in the sparkling wines. And here, because we've got a stepping stone of these compounds, depending on how much we've got, I've just included the 0, the 20%, and the 40% sour rot. And on your left is the phenolacetic acid, and you'll see you're climbing up now. And although at 40% sour rot, and actually even at the control EC 0%, what we see is we're still above the level of detection of this compound. It just gets more and more as you creep up. And a similar situation with ethyl phenol acetate, this nice compound, you've got those stepping stones. The thing about this is the level is so low. But again, this was taken after disgorging, and it will be interesting to see what happens during aging post-disgorging, because the levels of that nice honey compound are so incredibly low. There's still potential for that to change, but it's dependent on how much of phenolacetic acid is in there. So for sparkling, it doesn't seem to be too much of a problem, but we're going to have to see what happens with the sensory later on down the line to really ascertain that, because as I said, they're not the phenolacetic acid is at the level of detection, but they're not at the levels of consumer rejection. Let's just have a compare that to the red wines in 2020, which is a sour rot year. You can see there on the left that with EC 1118 in 2009, again, there's those stepping stones. There's a significant difference between them both. But that was because, 2019 was the botrytis year. And this is a really interesting thing that we didn't expect, because in 2020, where it's sour rot, we have much lower levels of these compounds. So it may be that in our cool climate that there's much higher levels of this horrible dusty, musty aroma in botrytis than what we were expecting, which was what it was going to be in sour rot. We'll have to have a look, because we'll have to also look at things like the amino acid, phenylalanine in the beginning of the juice and things like that we can have a look at. Okay, you'll be pleased to know, coming to the last and final slide. So we've completed all the work on Pinot Noir red wines for both years of the winemaking, and still analysing all that data, as you can see from the amount of data that we collected. The sparkling wines from 2019 and 2020, well, we finished all the laboratory analyses for 2019, and the sparkling wines for 2020 were just disgorged in last month in May at Millesy. And analysis is underway for all those wines at the moment, so we can complete that set later on this year. There were many people involved in this work all the way along. So Xiao and Jeff Moot, my colleague Wendy, I've got Debbie there, I thought I'd just better check, need to make sure she's there. Gary Pickering and Stephanie for the lab, and Hannah for our sensory work. And Jane Kelly and Leah, there's many of us involved, and Rachel there. The initial sensory work that was done to look at detection levels and consumer work was done by Amy Bowen and Amy Blake at Vineland Research and Innovation. And of course, all our funding partners and the wineries who are so often and so keen to work with us. So Andrew Peller and Hubel and Malcolm Laurie at the Laurie Vineyard. And there's some references for you there. And questions? I'm all finished.
Perfect. Thank you very much, Belinda. We haven't had any questions come through specifically for you right now, but I will invite all of our guest speakers to turn your cameras and microphones back on. And a reminder to our attendees as well, we have a few minutes now for a Q&A period. If you've got any questions, feel free to type them into the Q&A section at the bottom of your Zoom screen there. I went through a few of the slide presentations in advance, along with a couple of my CGCN colleagues, and drafted a few questions. So I've got some that I'll start the Q&A session off with, just for a few questions for Debbie, actually, and feel free as well, I'll say to any of our panelists, if you have questions for each other or you want to piggyback off someone's response, by all means, I'm happy to treat this as a bit more of an open forum discussion, if you will. So for Debbie, a few of my colleagues had questions for you about your presentation. First one here, how can viticulture techniques lead to different levels of skin tannins, for example, yield, sun exposure, or nutrient considerations?
Yeah, so we didn't do any of that work in this study, but work that Belinda's been involved with in the past. You know, certainly sun exposure is a good thing in terms of building up tannin. Some of those phenolic compounds, those flavonoids, there's many different classes of them. And some of them actually, you know, act as kind of sunscreen, you know, to the grapes. So if you expose them to sun, they'll start to build up, you know, that profile a little bit better. And that, of course, also helps with anthocyanin development as well. Whereas if we keep them shaded for too long, or you know, only take the leaves off at later in the season, they just don't have time to react properly, and you can sometimes do more damage than, you know, than good from what you were trying to accomplish. So, you know, I think getting, you know, good airflow helps reduce disease pressure. Getting the sunlight in will help, you know, with the tannin development. And for red wine production, that's what you want. You know, now, you know, I'll just qualify this, that, you know, Belinda has also been doing work on this for sparkling production. And that's a different scenario, because you don't want that, you know, extensive tannin development in your, you know, say Pinot Noir red grapes for sparkling production, because that's going to cause some bitterness, you know, in astringency in the wines, in the base wines. So, you know, you really have to match the wine style that you're going to be producing with the grape variety, you know, which I'm sure a lot of growers and winemakers know, but, you know, it's not one size fits all, you know, for all wine styles that we're looking to produce.
Right. Yeah. Yeah. Belinda, do you have any follow-up comments regarding the same question, but for sparkling production specifically, or did Debbie kind of cover that pretty well?
Yeah, I think Debbie's covered that. I mean, I should just add that we are in the process of finishing studies with regards to leaf removal and things like that for sparkling and red wines, both Cabernet Franc and Pinot Noir. And we're hard at work at those, and we'll be picking that up again really soon to finish and complete that. And that will provide information to the Ontario growers and winemakers with regards to the different styles and when to do it and when not, and how much in much more detail. So one to watch out for.
Great, awesome, thank you. Another question that we had from my colleagues, Debbie, you mentioned this early on in your presentation. Considering the point about LCBO looking for ripe, rich, rounded wines, has there been any research completed on how the structure, softness, roundness of tannin is impacted by the timing of harvest? Is it related to the level of extractable tannin?
Well, I mean, we haven't done those studies, right? And ripe, rich, rounded, you know, is a general description that LCBO is giving us, that, you know, we are interpreting a large component of that profile is going to come from tannin. You know, if you have too much harsh tannins extracted into the wine, your wine's going to be bitter and astringent, and it's not going to give that smooth silkiness, you know, that consumers are looking for. So, sorry, Ethan, I've lost my train of thought. The first part of, can you just repeat that question? Because there was a point I was going to make.
Yeah, absolutely. Yeah. So the LCBO is focused on the ripe, rich, round. Has there been any research completed on how the structure or softness or roundness of tannin is impacted by harvest timing?
Right. And so I think, you know, one of the difficulties that we have in tannin research is matching up tannin measurements in the grape to tannin values that result in the wine. And typically they don't marry very well. But many of the older techniques that we had for tannin measurements, you would just grind up the entire berry, okay? And so you're grinding up the skins, you're grinding up the seeds, and that, of course, is going to lead to much more tannin extraction, because you have much more surface area that's released that, you know, you can extract the tannin from. When we developed, you know, the method that we're using, Belinda put a lot of thought and time into how we were going to try to marry our extraction process to more mimic what was going to happen in the wine. So hence we painfully tease apart, the skin and the seeds from each other. And, and then there's some, a level of ethanol that's included in the extraction buffer, so to speak, during that extraction process to try to mimic what's happening in the wine. So we're hopeful, and again, we can't give final answers to this because we're still doing this work, but we're hopeful that the tannin measurements that we're generating now with the MCP assay under the conditions that we do the tannin extraction are going to be a better indicator of the resulting tannin, that can result in the wine and the profile that you're going to get in that wine. Unfortunately, COVID reared its ugly head for the last 2 years, and we've been severely limited on the sensory evaluation of these wines. We just weren't able to do them because of the COVID restrictions. So I can't even, for most of the trials, that we've carried out, we can't even give preliminary results other than our own tasting of the wines, which, isn't really fair to do. So hopefully by, this time next year, we'll have all of that sorted out and we'll be able to give a better answer to that question, because really at the heart of it, that's what we're trying to do. We're trying to provide a better measurement tool to the winemaker and the grower for when to harvest these grapes, and in terms of the tannin profile, and get that tannin measurement in as a regular measurement that's being done to help make those harvest decisions, and then look at ways to make use of that information to optimize the tannin extraction in the wine. So for instance, if you already have high skin tannin and low seed tannin, then you don't have to worry so much about your extended contact with the seeds, because you're already not extracting as much tannin from the seed, because typically those grapes are riper, they've got a harder seed coat, the seeds are more brown, you can physically see it, you're not going to extract as much. It's when the opposite scenario occurs, and you need to know that and be aware of that prior to going forward on your winemaking technique that you're going to use.
Right. Yeah, it's a very interesting concept. One final question for you here. Do these measured levels of tannins correlate with traditional sensory or tasting methods? In other words, could levels of extractable skin and seed tannins be reliably assessed by tasting grapes in the field?
Good question. And we actually didn't do that as part of this study. So at least not in a scientific way. So, that may be something for a future project, now that we've got this tannin base built up and we've got our designations, to try to see sensorially if we get that same perception, if we can differentiate that low, medium, and high, level of skin and seed tannin in the field compared to what we're seeing, upon the measurements that we're doing.
Great. Yeah, no, that's good to know. You can thank Ross Wise for that question.
Yeah. Great question. Yeah.
Perfect. Thanks very much, Deb. I've got a couple questions for Gavin and Lihua here. Either of you feel free to comment. I think they're a bit more tailored to Gavin's presentation. Do non-fermentative yeasts outcompete common spoilage microbes in must? Is the first question I have for you.
Gosh, it's an interesting question. I think they must. I'll use that word a different way. They must, because we're not seeing a lot of spoilage. We've got an awful lot of fungi, an awful lot of yeasts in there. Very high diversity in the must, but we're not getting spoilage. We do see probably some bacterial spoilage in one or two of the fermentations, but that's relatively rare. So yeah, I would think that they do have some. Actually, thinking back to the literature, they do have some biocontrol properties.
Great. Okay. Thank you. This one, I know you talked a little bit about, you know, different yeast strains that are found during or in the must and then in the final wine. But is the population of various yeasts being analyzed at different stages of the fermentation process as well?
No, we didn't track them in that much detail. We looked before and after. We didn't do a time series.
Gotcha. Fair
But
enough.
on the other hand, we were looking at 8 vineyards. So it was a lot.
Yeah, you can yeah, thank Ross again for that difficult question. He seems to be throwing all the hard balls here today.
Maybe I can add in a little bit.
Yeah.
And I know for Gavin's East communities analysis, and he collected initial sample and after the fermentation, but we did the monitor for the yeast and total population and the non-Saccharomyces yeast population, total bacterial load, lactic acid bacterial load, that is during the fermentation. So every 2 or 3 days, we take sample to monitor the microbial change. So we do have some data for that.
Okay, great, awesome. Yeah, I guess that's the nice thing about having 13A and 13B components to your project, is that a lot of the research is complementary. So that's great to know. Thank you. Another question that might be a little tough to answer, but I'll throw it out there anyway. How do the Indigenous yeast communities in Nova Scotia differ from those in other wine producing areas of Canada, such as Ontario, BC, or Quebec?
Do you want me to answer that, Lihua?
Yes, go ahead. Yeah.
Okay, so there haven't been a lot of these really thorough assessments of vineyards. This is the first one for Atlantic Canada. The only other one that comes to mind is from British Columbia. And what is interesting is that they also found a very high proportion of Saccharomyces uvarum. They found that Saccharomyces uvarum also dominated in BC vineyards. We're finding the same thing. So it seems to be a cool climate phenomenon. Maybe it's a Canadian phenomenon.
I could just add to that, Gavin, not that we've done the types of extensive studies that you have, but you know, we do have local wineries interested in cultivating their own, you know, yeast strains, and we've found it not difficult to isolate Saccharomyces uvarum strains from you know, from their ferments. So I would echo that. You know, there's certainly you know, it's a common species, you know, that we're finding in the non-inoculated ferments. You know, not done under the same strict conditions that you have, you know, but still, you know, we're picking them up, and you know, they're not using them in the winery, so you know, they must be coming from you know, from their vineyard.
Good.
Awesome. Yeah, thank you for the insight. Just one final question or thought that I had, I guess, from both Gavin and Lihua's presentations. You talked about part 1 and part 2. You mentioned part 3 of your overall project is some ongoing work. Do you care to, or are you able to elaborate at all on the ongoing work there?
So that the ongoing work is the actual fermentation with indigenous yeast. Maybe Lihua wants to answer that. I'm not sure.
Yes, we identified some good non-Saccharomyces yeast and Saccharomyces yeast. So after the characterization, we do selected some promising yeast. So this year we are put a lot of work to do the mixed and the sequential fermentation. And with that, we will add in some sensory evaluation. We do have a collaborator from different university. They will contribute to do the sensory work for that. So that is the last year for the class project. We will focus on the mixed and the sequential fermentation.
Great. Yeah, it's really good, you know, personally, to see that all of these projects have some really interesting results coming down the pipeline soon in year 5. So it's really great now that we have the update 4 years in, and obviously COVID has slowed some stuff down, as Debbie alluded to. But yeah, I think year 5 will be a really productive and really interesting year for all of these projects. That's really great to see. Before we wrap up here, I just really quickly need to obviously do some acknowledgements and some thanking of people here. So Obviously, special thanks to our presenters today. Debbie, Gavin, Lihua, and Belinda, your expertise is invaluable as always, and really appreciated you taking the time to put together the presentations and to share your knowledge with us here today. I know it's a busy time for a lot of people, so thank you for taking the time to be here with us today. Also, just for Lihua's presentation, legally to ensure we can post the webinar on our website, I just have a bit of a Creative Commons license attribution here for you. Just got to show that and we can move on. And that's all I have to say. Thank you for joining us. This is the end of our 8th and final webinar of 2022. They were extensive and diverse in topic, which is really, really great. And as I mentioned before, they'll be available on our YouTube channel and on our websites until the internet stops working, I guess. So if ever anybody wants to go back through, obviously I mentioned this webinar along with all the other ones have been recorded. So feel free once it's been posted to view it again or send the link to anybody who wasn't able to make it today. And you can keep up with CGCN on social media, through our bi-monthly newsletter, through our website, to stay up to date with the continuing research for all these cluster updates. Sometime late this year, we'll be continuing to plan for next year's webinar series as well, so you can keep up to date on that. We're always trying to expand upon our KTT efforts in that regard. So never a dull moment with us. You always have something to keep up with, which is really great to see. Once again, thank you to all of our presenters for being here with us today. Thank you for all of our attendees for joining us, and enjoy the rest of your day. Thank you so much, everybody.
Bye. Thank you.
Bye-bye.
you.
Thank you.
Take care.